Good morning, everyone. Take your seats, please. Okay, welcome to the second session of Cosyne 25, which is on circuit formation. We will have one invited and two contributed talks. The first talk is by Julijana Gjorgjieva from the Technical University Munich in Germany, and the title is Shaping Structure and Function via Synaptic Plasticity. Julijana?
Good morning, everybody. It's really nice to see everybody here today on the first day of the conference. I would like to take this opportunity to thank the program committee for inviting me to be here. It's really a great pleasure and an honor to be on this stage and to have the opportunity to tell you about some of our recent work. So already in very early stages of development, neural circuits undergo a huge restructuring of synaptic connectivity. With this change in synaptic connectivity comes the ability to perform different functions and behaviors. And this process of change, of course, continues throughout one's lifetime as we experience sensory stimuli and continue learning from prior experiences and context. This gives rise to neural circuits that are structured. This structure is reflected, first of all, in patterned neuronal activity, but also in non-random structures of connectivity. These have been identified by measuring the over- and under-representation of connectivity motifs involved in multiple cells in different brain areas, also simplexes or other graph structures, using various data sets, initially with multi-patch recordings and more recently with the availability of connectomic data sets. But simply knowing the topology of neurons doesn't mean that we can easily derive the structure that exists at the level of activity and connectivity. So what might be the mechanisms that play a role in the emergence of this structure? Broadly speaking, we can divide these mechanisms into two classes. The first class of mechanisms I call activity independent. So here we can think of maybe mechanisms with evolutionary origin, but I really mean those that involve the interactions of different sets of genes and molecules that determine where axons of neurons grow as they go from one area to another. These types of mechanisms have been most prominently studied in the emergence of sensory maps in sensory systems like the visual and the somatosensory. And here we see an example of these F and nephrin molecules whose interaction determines the emergence of the so-called topographic map in the visual system that's established between a primary sensory area like the retina and a downstream area like the superior colliculus. But there's a second class of mechanisms that play an equally important role in the organization of connectivity and activity, and those are these mechanisms that specifically depend on this patterned neuronal activity. So we call these activity-dependent mechanisms of synaptic plasticity. Of course, if you are looking at adult organisms, it is natural that there will be patterned activity simply by the fact that we experience the sensory world around us. But interestingly, already in very early stages of development and before the onset of sensory experience, many neural circuits in the developing brain can generate their own patterned spontaneous activity. I want to highlight for you some examples of this patterned activity that's generated in the developing visual system. What we see here is very strong spatiotemporal correlations that are seen in the propagation of spontaneous events in the developing retina that are known as retinal waves. I should note that these are recorded before the onset of sensory experience. These patterns of activity are known to propagate in downstream circuits. So here we see examples of so-called modular patterns, so they no longer look like these waves in areas like the primary somatosensory and the primary visual cortex. Now we know that blocking or perturbing this spontaneous activity can have very big influences on the emergence of organization in these neural circuits and resulting computations. So therefore, we can conclude that indeed these non-random patterns of neuronal activity somehow play a role, or a very important role, in the instruction and the refinement of connectivity in these circuits. So how can we go about studying these activity-dependent mechanisms of synaptic plasticity? Studying this process is a non-trivial challenge, and that's because activity and connectivity tend to influence each other in this feedback loop. The process is made even more complicated by the fact that neural circuits are recurrently connected. So it's difficult to investigate how local synaptic plasticity mechanisms, which are these biologically plausible mechanisms that we're interested in, really modify one connection when you have a fully connected recurrent circuit. Moreover, these mechanisms also depend or may differ across different cell types and different brain regions. They may be influenced by neuromodulatory signals, behavioral states, and importantly, they operate over different time scales. So saying that we are studying synaptic plasticity is a very broad term because synaptic plasticity encompasses all of these different types of mechanisms. On one hand, the changes in the efficacy of individual synapses through these short-term plasticity mechanisms such as the facilitation or depression of synapses. On the other hand, also long-term synaptic plasticity mechanisms that more permanently change the strength of the connections in these neural circuits. Here it's worth mentioning, of course, Hebbian plasticity or spike timing dependent plasticity. And there's finally a class of mechanisms known as homeostatic that operate on even longer time scales, such as synaptic scaling and intrinsic plasticity, that act to maintain or restore network function subject to external perturbations. So in my lab, we have been developing biologically plausible mechanistic models to investigate the operation and the action of these different forms of synaptic plasticity operating over these different time scales, to investigate the emergence of structured and patterned connectivity and activity that also change on these multiple time scales. And what I will show you now are some examples that focus on short-term and long-term plasticity. And I will start by asking the question of how this structured connectivity may emerge in recurrently connected circuits of excitatory and inhibitory neurons, where the connections are learned by exposing these circuits, by showing these circuits patterned inputs. So the goal here is to see whether this long-term plasticity can self-organize the circuits in structures that give rise to interesting dynamical regimes, whereby the circuits can perform interesting computations. And so here's an example of one such nonlinear computation that can be realized by these recurrent circuits. This is known as response normalization and was measured initially by Laura Busse in the cat visual cortex, but this has also been measured in the rodent visual system as well. This is a computation that basically characterizes the response of the population of neurons in the visual cortex to initially stimuli that are relatively simple, so just oriented gratings. So you see that the responses of the population here are tuned with a peak at the orientation of the shown stimulus. But importantly, when two of these stimuli are shown together in the form of this plaid stimulus grating, the response of the population is nonlinear, meaning that it's smaller than the sum of the individual responses together. So the network can do this sublinear summation. Moreover, if one of the stimuli is shown at a lower contrast than the other, then the network does a winner-take-all computation, meaning that the lower contrast stimulus is suppressed at the expense of the higher contrast stimulus, which is amplified. So here we wanted to investigate how this response normalization may emerge in biologically plausible neural circuits. And a model that has been proposed more than 10 years ago by colleagues like Yashar Ahmadian and Ken Miller that can perform this computation and several other interesting nonlinear computations is a network known as the stabilized superlinear network, or the SSN. This network has several defining characteristics that enable it to perform this computation. First of all, the individual neurons follow this nonlinear input-output relation. So if we look in the upper right corner, you see this power law-like function with an exponent that has to be bigger than 1. And second of all, the neurons, the excitatory and the inhibitory neurons in this network, have to be strongly recurrently connected so that the network is in a so-called inhibition-stabilized regime. This means that inhibition is absolutely necessary to stabilize the excitatory dynamics in this network. Besides this recurrent connectivity and inputs, neurons in this network also receive feed-forward inputs via these uniformly distributed tuning curves in input space, so that one can derive the population response of this network to a given stimulus, and it looks just like the experimental response with a peak at the orientation of the shown stimulus. Now, indeed, this network has been shown to perform this computation known as response normalization, whereby the response to this summed stimulus is lower, namely the summation is sublinear relative to the two individual responses together. Well, this is all wonderful. However, one caveat of these networks is that they have hardwired connectivity, meaning that the connectivity has to either be tuned or picked by hand, of course, using guidance from experimental data, or at best fitted using different optimization techniques to experimental data. We wanted to investigate whether these important long-term plasticity mechanisms that really, we think, play an important role in structuring connectivity can also realize this type of strong recurrent excitatory coupling in these networks. And so to do this, we investigated recurrently connected circuits of excitatory and inhibitory neurons that were initially weakly and randomly connected. And the important part is here that they were all subject to synaptic plasticity. So those of you who study plasticity, you know that tuning such a network, putting in plasticity rules that can generate stable learning and stable evolution of the weights, this is a challenging problem. So we wanted to investigate a very simple, very general form of long-term plasticity, and we picked Hebbian plasticity. So probably you all know what Hebbian plasticity is. It basically says that the connection between a presynaptic neuron Y and a postsynaptic neuron Z will potentiate subject to coincident activity of these two neurons. However, you also know that this Hebbian plasticity is a positive feedback process, so it's extremely unstable in the sense that the more the connection potentiates, the more the activity of the postsynaptic neuron increases in this positive feedback loop, and this is made more complicated if there are other recurrent interactions in the circuit. And so a former PhD student in my lab, Samuel Ekman, who is now a postdoc in Cambridge, proposed the solution to this problem of instability of Hebbian learning by introducing a normalization on the synaptic weights that experience Hebbian plasticity. So basically, a synapse can only grow at the expense of other synapses. So in this case, the middle synapse undergoes a long-term potentiation, and as a result, the network undergoes this normalization of the weights, whereby all the weights get suppressed while preserving their relative strengths. Now, this type of normalization has, of course, experimental origins, can be verified in experimental data, and so these are some classic studies from Bourne and Harris a long time ago that basically found that in excitatory synapses in the hippocampus, the total synaptic area of the synapses is preserved before and after the induction of LTP. So we can formulate this problem by basically looking at a very simple feed-forward system with an array of feed-forward neurons, Y, and a single postsynaptic neuron Z. And here we put in this Hebbian plasticity rule plus the normalization that maintains the sum of the weights at a given constant W. Now you all know, you are familiar with this plasticity rule. This is the well-known Oya rule, and Miller and Mackay studied the role of this normalization also very nicely 30 years ago. What is nice about this weight dynamics is that we can easily find a fixed point solution of the weight vector, and we find that the fixed point of this dynamical system basically lines up with the first principal component of the input covariance matrix. So this means that this plasticity rule, this Hebbian rule, plus the normalization, basically does PCA of the inputs. However, what Samuel noticed in this original study, that the same type of preservation of synaptic resources also applies to inhibitory synapses. So again, the total synaptic area of inhibitory synapses before and after the induction of LTP is also preserved. So he extended the problem by adding now a second population of inhibitory cells that project to the same postsynaptic neuron Z and assume the same kind of Hebbian learning at all the synapses and introduce an additional constraint for the inhibitory synapses. This is a different bound, so the sum of the weights is maintained at a different level because the excitatory and inhibitory synapses basically compete for separate pools of synaptic resources. And so studying this system is also very easy in this feed-forward setting where we assume linear rate dynamics. We can derive again the fixed point of the weight vector, and we see, although the covariance matrix is slightly modified due to the presence of the inhibitory inputs, we see again that the weight vector lines up with the first principal component of the input covariance matrix. It just goes in the opposite direction of the excitatory weights. Now, Samuel also simulated this in a network with a nonlinear postsynaptic neuron, because we're after generating organized connectivity in the SSN, which is characterized by these nonlinear neurons. And he found, again, the emergence of this excitatory and inhibitory co-tuning. So you see the tuning curves have exactly the same peak for the two sets of weights, also when he had this nonlinear postsynaptic neuron. However, all of this works really well in a feed-forward network. How can we translate any of these intuitions and analysis in a recurrent network? Before we looked at the fully connected recurrent circuit, we decomposed the problem into smaller connectivity motifs. So first we consider the connectivity motif that it consists of two excitatory cells, one which is plastic. This is the neuron in the dark blue. You see that it has a flat tuning curve as a function of the input, so the input weights are all the same with very slight noise fluctuations. And then we had a second excitatory input in this circuit, excitatory neuron, with a fixed and tuned tuning curve. So the input weights follow this Gaussian profile with a very clear peak. This static neuron projected to the plastic neuron with a recurrent connection that was also plastic. And so we saw that, subject to this Hebbian learning with this normalization constraint, the influence of these excitatory effects in the recurrent circuit was attractive, meaning that the tuning curve of the plastic neuron became the same as the tuning curve of the static neuron. We repeated the exercise by now having a motif that involves only static inhibitory neurons with static or fixed tuning curves and a plastic excitatory cell that receives these plastic inputs from the static inhibitory neurons. And here we saw the opposite. While the excitatory recurrent influence is attractive, the inhibitory influence in this recurrent circuit is repelling, meaning that the tuning curve of the plastic neuron actually ends up distributing itself in space to generate as little overlap as possible with the tuning curves of the static cells. We then complexified the circuit motif even more. Now we introduced two excitatory cells that both had tuned input weights, but they were plastic, so they were allowed to change due to recurrent interactions. And again, we saw this evidence of the excitatory recurrent plasticity that is attractive, that basically brought the tuning curves to converge onto the same one. And similarly, when we started with multiple inhibitory neurons in the circuit, we saw the repelling effect whereby the tuning curves ended up distributing themselves in space. So clearly, these inhibitory neurons can actually decorrelate themselves and generate tuning curves that are uniformly spread out in space. So next, we wanted to investigate whether these inhibitory interactions can also be used to decorrelate the excitatory inputs when we took into account the entire recurrently connected circuit where all the feed-forward and recurrent connections were plastic. So subject to this Hebbian plasticity learning plus these two normalization constraints for the excitatory and the inhibitory synapses, indeed we saw that the effects of these inhibitory neurons also get imprinted on the excitatory ones in the sense that the feed-forward tuning curves now of both populations end up completely decorrelating in space. So they end up filling input space with as little overlap as possible. We then looked at the emergent connectivity in the recurrent circuit. So here we looked at the connectivity matrix by arranging the neurons according to their feed-forward preference, meaning neurons that had a similar feed-forward tuning curve were placed next to each other. And what you can see from this connectivity matrix is the emergence of structure, namely this block diagonal structure. So the dark color here indicates strong weights. So what we see is the emergence of like-to-like recurrent connectivity, which again is found in experimental data. So finally, the big question that we're after. Clearly, we have a circuit that is organized in a way that's consistent with experimental data, but is this circuit really in a computational regime that can actually do something interesting? So indeed, we tested the ability of the circuit to do this response normalization computation we showed one stimuli at a time, and we measured the responses of the population, and we showed the two stimuli together, and we saw that indeed the network can do this sublinear summation of the inputs. And it can also do this winner-take-all competition by suppressing the weaker at the expense of the stronger stimulus. Now, I don't have time to show you this, but we were also able to investigate the emergence of other nonlinear computations in this network, namely surround suppression. So we kind of replicated the motifs, the circuit in the network, one to model the center and one to model the surround. And we showed different oriented stimuli to the center and the surround, and we saw that indeed the network can do this surround suppression, whereby if you increase the size of the stimulus that you show, you decrease the response of the network. However, besides this decorrelation of the tuning curves, this emergence of like-to-like connectivity, there is even finer scale connectivity structures that emerge in neural circuits at the level of specific connectivity motifs. I already alluded to this at the beginning. But here I want to focus specifically on connectivity motifs that have been found to involve inhibition. And I want to highlight a particular study here that found these motifs involving inhibition in the visual cortex, performed by Peter Znamensky, published last year, that measured the strength of the inhibitory connection from a class of inhibitory neurons, the provalbumin positive or PV cells, into excitatory pyramidal cells in the visual cortex. So what he found was that the strength of the connections from these inhibitory to the excitatory cells is proportional to the strength of the excitatory connections from the pyramidal to the PV cell. So if one is strong, the other one is strong, and vice versa. So together with a postdoc in my lab, Dylan Festa, we wanted to investigate the emergence of specific connectivity motifs that involve inhibition. And so here, rather than working with these general rate-based plasticity rules like Hebbian that I showed you in the first part of the talk, we wanted to see whether other factors beyond just firing rates, such as the spike timing of individual neurons, may also influence the emergence of different connectivity motifs. So first we looked at the simple EI motif comprised of one excitatory and one inhibitory cell, where the connection from excitation to inhibition was static and the connection from inhibition to excitation was plastic. And we investigated different forms of long-term plasticity rules that now take into account the spike timing. So these are forms of inhibitory spike timing dependent plasticity. Many of these forms have been measured experimentally in Anne-Marie Oswald's lab and Rob Fromke's lab, but here we wanted to, rather than pick one particular or another rule, we wanted to investigate the whole family of inhibitory plasticity rules and study their consequences on formation of these inhibitory circuit motifs. And so in this case, we considered a set of rules that we called anti-Hebbian, whereby if a pre follows by a postsynaptic spike induces potentiation, induces depression, so this is why we call them anti-Hebbian. They can be asymmetric or symmetric. And then a second class of plasticity rules that we call Hebbian, where if a pre follows a postsynaptic spike and induces potentiation, that's why because of the causality, we call them Hebbian. So what Dylan found in this framework, that the first set of anti-Hebbian rules are saturating, in the sense that if you rely on the spike timing covariance between the inputs and the outputs, these rules cannot actually generate connectivity motifs because they either saturate the weights or depress them to zero. But he found that the second class of Hebbian plasticity rules, both the symmetric and asymmetric, are self-stabilizing in the sense they can give rise to the emergence of these different motifs that include the inhibitory neuron. One is this blanket inhibition motif, where inhibition makes unselective connections with different excitatory cells. And then the other two, where inhibition makes specific and selective connections to the excitatory cells, depending on whether the excitatory cell makes a connection back to the inhibitory neuron. So mutual connectivity and lateral inhibition. So Dylan developed this theoretical framework that was actually initially developed by Kempter and Gerstner a long time ago for excitatory synapses. So assuming separation of time scales where learning is much slower compared to the dynamics, to the time scales of neuronal activity, he was able to write an expression for the mean change of the synaptic weight as a function of different features of firing of the neurons, namely the firing rates as well as the pre-post spike correlations or covariances, and the shape of the inhibitory STDP rule. So in this decomposition, There are 4 terms. The first 2 specifically depend on either the pre or the postsynaptic firing rate of the neurons. They're scaled by 2 constants, alpha, that can be set by us as the experimenters here. And there are 2 other terms that depend on the joint activity of the pre and the post neurons. The third term here depends on the product of the pre and the postsynaptic firing and is scaled by a constant that we call B that specifically depends on the shape of the plasticity rule. So B is given by the balance of the LTP and the LTD components in the STDP kernel. So for the rule that you see here, you see there's an equal amount of red and blue, meaning that the LTP and the LTD are balanced. So this would result in a factor B equal to 0. But if LTP dominates, then B will be positive. And if LTD dominates, then B will be negative. And finally, there's a 4th term in the equation that specifies how the spike covariance of the inputs, together with the shape of the STDP rule, contributes to the mean change of the synaptic weight. And so what Dylan found that basically by trading off different aspects of these different terms in the equation, one can determine the emergence of different motifs involving inhibition. So first of all, if we allow the first and the third term to dominate, by picking a plasticity rule where the term B is big, so here rate-based influences, so rate-based interactions dominate over the covariance-based contributions in the 4th term of this equation, then we see the emergence of this blanket inhibition motif. And this was actually exactly the plasticity rule studied by Vogels and Sprekeler in their science paper in 2011. So this generates sort of unspecific inhibition and firing rate homeostasis. So all excitatory cells reach a certain target level of firing. However, if we choose for other terms in this equation to dominate, in this case, if we choose plasticity rule, where this rate-based factor determined by B, which is the balance of LTP and LTD, is 0, so we choose plasticity rules and that have the same amount of LTP and LTD, then the 4th term that depends on the input-output spike covariance dominates determines which motifs emerge. And here the shape of the rule really determines the motif. So if we have this symmetric rule, we have the emergence of the mutual connection motif. And if we have the asymmetric rule, we have the emergence of this lateral inhibition motif. And finally, we wanted to see whether this intuition from these motifs translates also to larger networks. And before I tell you what we did, I just want to highlight some work done by Brent Dorion and Feresh Delagzi and Anne-Marie Oswald that actually found this lateral inhibition motif to be very important for the separation of assembly structures, which are groups of strongly connected neurons in large-scale sparking networks. So we also built our large-scale spiking networks and wanted to see whether the types of plasticity rules that we put can really translate at the level of these larger structures. We aligned or we organized all of the neurons along a one-dimensional ring where we preceded excitatory connectivity, but we assumed that the connections from the inhibitory to the excitatory neurons were plastic. We assumed here 2 classes of inhibitory cells that basically follow different plasticity rules when projecting to the excitatory cells. So we have the pink class that follows the symmetric rule, and we have the orange or yellow class that follows this asymmetric rule. Now, in Anne-Marie Oswald's lab, they measured and they indeed found these 2 different rules to basically be at synapses from 2 different inhibitory classes. The PV2E, so these are the parvalbumin positive neurons that I already mentioned, seem to follow this symmetric plasticity rule, while another class, the somatostatin positive interneurons, when they project to the excitatory neurons, follow this asymmetric plasticity rule. So from now on, we can identify our 2 inhibitory classes with these 2 PV and SST populations. And if we assume or if we precede excitatory to excitatory connectivity to have this distance-dependent profile where nearby neurons connect more strongly than faraway neurons, and if we assume the same for excitatory to inhibitory connectivity, but now put in the plasticity from the different inhibitory populations to the excitatory cells, we see that our intuition that we could study analytically for the small motif also translates in these large-scale networks. Namely, the PV to E connectivity becomes co-tuned to excitation, so it has the same profile, just opposite in sign, and the SST to E connectivity acquires this lateral inhibition. We could then do a simple linear response theory to quantify the effective weight in this network for the excitatory cells. And we found that this basically generated a profile of connectivity that looked like a Mexican hat, meaning strong excitation in the middle that was flanked by inhibition. And so given that we see the emergence of structured connectivity, we then stimulated the networks with unstructured input, and we saw the emergence of resulting structured activity as well. And to quantify the structured activity, we measured the Pearson correlation between cells as a function of the distance, and we saw this reverberating correlation curve that is basically telling us that there's long-range correlations that emerge in these networks. So we could then show the network stimuli with different sizes and show that the network can even do other types of things like this surround suppression computation, whereby as the stimulus that is shown to the network increases in size, the network shows a reduced response. And this is due to the recruitment of this lateral inhibition from the SST cells. And so in the last part of the talk, I want to switch gears a little bit because everything that I've shown you so far has dealt with mechanisms of long-term synaptic plasticity, whereby depending on the rule that we choose, whether it's rate-based Hebbian or these different forms of spike timing dependent plasticity, can give rise to changes or more permanent changes in connectivity that can change the structure of networks. But you all know that circuits can change also their dynamics, their activity patterns on a much faster time scale. So you don't really have time to wait for long-term plasticity to modify the connections. As we see sensory stimuli, and there's also top-down signals that can change this, such as changes in behavioral state. And here I want to highlight locomotion as a very prominent behavioral state that can change the firing of cells. So to understand how this happens, we wanted to take a look at other types of plasticity mechanisms. And to do this, we built a bigger or more complex biologically inspired circuit by adding the final class of inhibitory interneurons, namely the VAPs, that make up the classical canonical microcircuit model. So together with the SSTs and PVs, these three classes make up about 90% of all inhibitory cells in the cortex. And we wanted to add this VIP class because they are actually seen to be mainly responding when animals transition to locomotion. And as I said, we cannot wait to see these fast changes in activity. We cannot wait for the circuit to be modified by long-term plasticity. So we wanted to investigate how other mechanisms of plasticity that on a very fast time scale change the efficacy of synapses, such as short-term plasticity, may give rise to interesting dynamical regimes and computations. So here we actually considered measurements of short-term plasticity performed at the Allen Institute. I believe Luc Campagnola presented them a few years ago at this very same stage, where they measured specifically forms of both facilitation and depression at different pairs of synaptic connections in this microcircuit. And the computation that we wanted to investigate here had to do with how changes in both the bottom-up inputs, meaning the sensory environment of the animal, and top-down influences, such as changes from stationary to locomotion, on a very fast time scale change the firing of the neurons that are involved in the circuit. So several experiments here showed that under conditions of darkness, if the animal transitions from stationary to running, the SST population firing decreases. On the other hand, if the experiment is performed under conditions of visual stimulation, where the animal sees gratings or other types of stimuli, then the SST population is increased, with everything else in the circuit being the same. So we wanted to investigate how a network with these multiple interneuron classes and the excitatory population, in the presence of these short-term plasticity mechanisms, can give rise to this response reversal of SST. And here we actually considered, rather than all of the STP mechanisms measured experimentally, we considered the four most prominent ones. Three are short-term depressing plasticity mechanisms where the PV is the originating cell, and the last one is a short-term facilitating mechanism from the SST to the VIP neurons. So this was work done by a current PhD student in the lab, Felix Weizmann, and a former PhD student, Chris Wu, who is now a postdoc at Columbia. So what they did is they investigated the emergence of this response reversal by changing the bottom-up input to the network. This was provided to the E and the PV population as a measure of this visual environment of the animal, while applying top-down modulation to the VIP neurons, which are known to increase their activity during locomotion. So what they found is that indeed, in this low baseline case, the circuit shows this, in this case, this decrease in the SST firing. But if we look at the same type of behavior in a circuit in a high baseline state, we see that the SST population firing increases. So this is exactly the response reversal that we saw in the experimental data. So this circuit equipped with these short-term plasticity mechanisms can capture this response reversal. But what might be the mechanism behind it? And is it really due to the short-term plasticity that we put in the circuit? So to answer this, we eliminated the short-term plasticity mechanisms, and we found that the circuit can no longer do the response reversal of the SST cells. We then wanted to investigate specifically which of the 4 short-term plasticity mechanisms that we put in, the 3 depressing and 1 facilitating mechanism, is responsible for this reversal. And what we found is that the one that was most important for the reversal was the depressing, short-term depression mechanism from the PV to the excitatory population, which was a little bit counterintuitive to us because the synapse is actually far away from the population that we're modulating, which is the VIP, and the population that is changing the response, which is the SST population. So finally, we wanted to see how, due to these mechanisms, the dynamical regime of the network changes. Because if you really think about it, I alluded to inhibition stabilization earlier in my talk, the type of response that we see in the SST cells, where they really reverse their firing up or down, depending on whether you're in the darkness or in the light, is sort of reminiscent of a dynamical effect known as the paradoxical effect that people have studied as a dynamical signature of inhibition-stabilized networks, most prominently just networks with a single excitatory and a single inhibitory population. So here the paradoxical effect simply refers to applying a perturbation to the inhibitory cell, a positive current into the inhibitory cell, and seeing the response of this population basically decrease. So you drive the population, but the population decreases. This is what is paradoxical about it. But this is perhaps not so paradoxical if you realize that these networks that show this effect are operating the inhibition-stabilized regime, meaning that you have this very strong coupling between excitation and inhibition, and that's why you see the effect. And so indeed, we applied the same perturbation to the SST population here to see whether the response reversal computation that we see is related potentially to a paradoxical effect, and we found that to be the case. In fact, so in the low baseline case, applying the perturbation increases the SST firing, so this is not paradoxical. You apply a drive and you see the population go up. But in the high baseline case, so for high bottom-up input, when you drive the SST population, you see that the firing goes down. So you see the emergence of the paradoxical effect, and we could demonstrate that these two are actually mathematically equivalent to each other. And so finally, I want to tell you about the relationship between the paradoxical effect and the inhibition stabilization of these networks. Because I already showed you for these EI networks, the paradoxical effect is used as a dynamical signature to determine whether the networks are in the inhibition stabilized regime or not. So to investigate this in our circuits, we wanted to see how the response reversal of SST relates to inhibition stabilization. So to do this, we selectively eliminated different interneurons in this network by freezing them, and then investigated if the resulting subnetwork without the frozen interneuron has stable or unstable dynamics. So first, we froze the SST population, and we considered the dynamics of the remaining E, PV, and VIP network. To do this, we computed the leading eigenvalue of this subnetwork, and we found that the leading eigenvalue transitioned from being negative to being positive at some value of this bottom-up input. In other words, for these regimes of high bottom-up input, so remember these are the regimes of visual stimulation, we find that the SST is absolutely required for the network to be stabilized. But what happens in the low bottom-up input case? So here we did the same thing by freezing the PV population and investigating the dynamics of the remaining network, which excludes PV, so the E, SST, and VIP subnetwork. And here we found that the eigenvalue of the leading eigenvalue of the system is always negative. So what this means is that this low bottom-up input regime either PV or SST can stabilize the network. And I should note, just for completeness, that the VIP cells do not play a role in the stabilization of this network. Okay, so I will now go to my summary slide to basically tell you what I told you, that we included, we built examples, I showed you examples of building biologically plausible mechanisms at different levels of complexity that include different classes of interneurons and mechanisms of synaptic plasticity that operate over these different timescales. And I showed you how these can, if we look at the role of these long-term synaptic plasticity mechanisms, these can really change the structure of these neural circuits by changing the strengths of the synaptic connections over these longer timescales and give rise to these interesting motifs and computations. But also on a faster timescale, these mechanisms of short-term plasticity can really very quickly change the dynamical response regime of these networks without waiting for these long-term plasticity mechanisms to induce synaptic change. And finally, I would like to thank my lab. It's really been wonderful to talk to them, and it's always inspiring with bringing lots of new ideas, and especially the people who did the work, so Dylan, Felix, Samuel, and Chris. And indeed, we broke the pillow rule, so I encourage you to go and look at some of the posters tonight, today, and tomorrow. Unfortunately, the fifth presenter couldn't make it due to visa issues. But yeah, thank you very much for the attention, and happy to take questions.
Yeah, we have time for a few questions.
Nobody's awake.
Okay, maybe I can ask you a question.
Sure.
So in the first part of your talk, you have shown that you can get these inhibition stabilized dynamics. So I was wondering whether you can also get other types of, let's say, for example, bump attractor dynamics, or under which conditions you would get networks of different dynamics, right, with kind of similar rules, or what need to be changed?
Yeah, so actually in the first part of the bump attractor dynamics can actually be generated with these inhibitory plasticity rules that I showed, I think, in the second part of the talk, by including the symmetric and the asymmetric Hebbian rules at the synapses from the PV and the SST cells back to the excitatory population, because this is where, you know, we assume this ring structure. We line up the neurons organized in this one-dimensional line. And then through plasticity, we see the emergence of this Mexican hat profile. And these networks, these ring networks with this Mexican hat profile have indeed been studied classically to generate this continuous attractor dynamics and the persistent bump of activity. And so we can indeed generate these in our network, so I didn't show this. So this emerges really spontaneously by putting in the right plasticity rules in the network. Yeah.
Okay, we have a question here.
Yes, hello, great talk. Two short questions. So one, when you look at the plasticity in these inhibition stabilized recurrent networks, I noticed that you see tuning curves that are spread out in terms of their preference, right, across
Yes.
orientation space.
It fills input space, yes. Mm-hmm.
Yeah. Do you also see, depending on the structure of the network, changes on the width of or shape of the tuning curve in addition to the preference?
Yes, so yeah, to fully analyze the circuit, we can linearize it, and we can do some interesting studies. So as long as in each of these units, let's call them excitatory and inhibitory units. So inhibition here is important to decorrelate the excitatory tuning curves, right? And so we can think of having separate excitatory and inhibitory units that are tuned to each of these input modes that correspond to the tuning curves that are spread out in space. So as long as each unit has enough inhibition, they will indeed be spread out in space with equal tuning. But if we start messing up the numbers, so if we don't have enough inhibition in some of the circuits to decorrelate them, then we can indeed see some broadening of the tuning curves. I know that Samuel has continued to think about this by basically exposing these networks to learning subject to natural stimuli that also contain edges and other effects that are then processed by Gabor filters before they get to the neurons. And I think there's some interesting effects that emerge there.
Awesome. Second short question. So you also mentioned you see this structure of activity in terms of spontaneous in the network.
Yes.
Is there a relationship between the structure of that activity and the representation that emerges as organized across the network?
That's a very good question. So I didn't note this, but as I started telling you in my talk, a large part of the work in my lab has been built on looking at synaptic plasticity mechanism in the context of development. And we were very interested in the emergence of these structured patterns of activity because they really look like a certain set of patterns seen in the developing visual cortex of the ferret. So these are studies by David Fitzpatrick, for example. And yeah, and so this is indeed very interesting, and we're continuing this. I was hoping to maybe have a cool video to show you of how this matches these activity patterns in the cortex, but yeah.
Yeah, just ask us for it. Thank you.
Yes, perfect. Let's talk after.
Let's take one quick question from the other side, please. And maybe the next.
He actually stole both my questions, so go ahead. Okay, so in addition to Hebbian mechanisms, you briefly mentioned homeostatic mechanisms. I wondered if you've worked out a satisfactory interaction circuit model that would incorporate both in a way that you think is realistic.
Yeah, so we have a few studies where we have also looked at the role of these homeostatic mechanisms that, I mean, the idea is always, okay, they operate over these long time scales, and so they're not really used to quickly control the dynamics of neural networks. For this, you need more faster mechanisms like inhibitory plasticity, which you could also think of it as being a fast homeostatic mechanism. But we have some studies where we have put in intrinsic plasticity or synaptic scaling and investigated how assaults on the network in the form of, let's say, stroke or sensory perturbations like monocular deprivation can give rise to changes in tuning very quickly, and then with the onset of these plasticity mechanisms, their re-emergence, or not, right? Because we know there's disease, and so sometimes these mechanisms fail. So this is very much also another interest and ongoing work in the lab.
Okay, let's thank Julijana again. And our next speaker is Samuel Muscinelli from Columbia University. And the title of the talk is Experience-Dependent Connectivity of Inhibitory Neurons in the Olfactory Cortex. Samuel.
Okay. Since we're in Montreal, my experimentalist colleague went to collect some data to compare the city's two famous bagel rivals. And no offense, but they couldn't really tell them apart. But with experience, locals, they start to notice the subtle differences. And before you know it, you developed an opinion so strong that even a newspaper more than 300 miles away writes about it. Perceptual learning is the term that is used to describe this phenomenon, in which by experience we come to better discriminate the features of our environment, and we get to argue about almost identical bagels. Perceptual learning has been shown in a multitude of different species and sensory modalities, including in olfaction in rodents, and it doesn't require feedback or reinforcement. So we set out to find out whether experience reorganizes the rodent olfactory circuitry in a way that could increase olfactory acuity. Here's our experiment. We let mice in their home cage smell a set of 4 neutral odors without rewards, hundreds of times per day for 2 weeks. I would call them the experience mice. Then, we performed acute electrophysiological recordings in the olfactory cortex, also known as the piriform, and measured the responses without any task to 2 sets of 4 odors each, the familiar set and 1 novel set that the animal had never smelled before. And finally, we assessed odor discriminability using population decoding. And what we found is that the responses of the piriform cortex to these 4 familiar odors were more easily discriminable compared to the novel odors. And this shows the classification error. And the reason for this improvement is that the responses were also higher dimensional, decorrelated. Now, crucially, both of these effects were experience dependent. In a new cohort of mice that did not have this prior experience, we did not see them. So piriform activity becomes higher dimensional and more discriminable. And this has been previously shown in the olfactory cortex as well as in other brain regions. And in fact, this afternoon we'll hear another example of this from Wayne and Son, who will describe his work on decorrelation in the hippocampus. Now, it seemed reasonable to assume that these experience-dependent changes in the activity of the piriform might be due to changes in the underlying circuitry. So we asked, is the piriform connectivity also reorganized in a way that could decorrelate the responses? To answer this, we needed a way to measure both activity and synaptic connectivity. Now, measuring synaptic connectivity in vivo is very difficult, but the piriform cortex presents an opportunity to solve this. It is a recurrent network whose synapses are plastic in a slice, and many in the field have viewed the piriform as a learning system for olfaction. To measure both the activity and the connectivity, we developed a new strategy to get into the old problem of inferring synaptic connectivity from pairs of spike trains. Now, those of you who are familiar with this, you know that it has a fraught history, but you also know that sparse connectivity helps, because it mitigates many confounds and in particular common inputs, which have hindered this approach for the past 50 years. Fortunately for us, the piriform is remarkably sparse. Its neurons are estimated to be connected, even locally, only around 0.1% of the time. So to find these rare connections among the large number of possible pairs, we developed a deep convolutional network that identifies spike time cross-correlograms looking like this one. So for each pair of neurons, this curve describes one neuron's firing rate relative to when the other one spiked. In this example, we see the signature of a connected pair, an asymmetric peak that reflects the underlying EPSP. It has a fast rise at a couple of millisecond latency, followed by a slower decay. And our convolutional network is able to look at all these cross-correlograms and recognize this characteristic asymmetric shape precisely. And here are some examples. And when it does, I'm going to call it a likely monosynaptic connection. Now, this is a pretty bold claim to make, but here's why we feel comfortable making it. So first, we validated our method using a spiking network model with known synaptic connectivity on which it was able to identify connections with 98% precision, which compares favorably to the state of the art. Our performance is in blue. Then, we estimated our error rate on our own recordings using several independent analyses. For example, we asked how often we detect an excitatory synapse coming out of an inhibitory neuron. And collectively, this analysis indicated when we infer a connection, we are wrong less than 5% of the time. And finally, we used the gold standard, an in vivo ground truth data set from Sam McKenzie in English with positively identified synaptic connections. On it, our deep network did not get fooled a single time, although I should mention that this data set only has around 30 identified connections to work with. So our method is precise. If it says a pair of neurons is connected, it is probably right. But it is also conservative. In the trade-off between precision and recall, we chose to be in this region with high precision and moderate recall. And in our data, we think we are recovering about 30% of the connections between the neurons that we are observing. So here's an example data set based on a single recording. Black points are an estimate of where the neurons are relative to the probe. Red lines are the connections we have inferred. So I'm flying over all this because of time, but we also looked at putative inhibitory connections using a separate deep network that we trained and validated using a similar strategy. So the bottom line is, although inference is challenging, our new method combined with the piriform sparsity gives us a reliable estimate of a subset of our connections. There's a lot more under the hood. We detailed this exhaustively in a recent preprint. I'm going to go back to the experiment now, but please come find me after. So for both the experienced mice that had these 2 weeks of familiarization with odors and the naive mice that did not have this experience, we measure connectivity using several hours of spontaneous activity, and during a separate phase of the same recording, we measure the responses of the same neurons to the 2 sets of odors. So now we can finally ask, what effect did this experience have on the network connectivity? Of course, at first we looked for signatures of Hebbian plasticity. And similarly to what has been observed in V1, we found that neurons with similar tuning have enriched connectivity. And this plot shows that the connection probability of 2 neurons increases with the correlation of their trial averaged responses to the familiar odors. So this was initially really exciting for us because it looked like fire together, wire together. However, we also found the same like-to-like connectivity for novel odors that this animal had never smelled before, and we found it even in naive animals. So at least within the kind of passive experience and time scale that we considered, we found no evidence of a Hebbian mechanism enhancing like-to-like connectivity in the adult piriform. And actually, this is a good thing, because Hebbian plasticity between excitatory neurons will tend to correlate the responses. And remember, we are trying to distinguish these two bagels. We're not trying to make one giant everything bagel. So if instead you want to decorrelate, you might want to work on inhibition. So we took a close look at inhibitory neurons, by which I mean fast-spiking neurons identified by their waveform. And for this example, inhibitory neuron here in blue, we found one excitatory input and one inhibitory output. But when we looked at this other inhibitory neuron, we found many more inputs and outputs. And so we wondered whether there was some feature of how an inhibitory neuron responds to the familiar odors that can predict how connected the neuron is. And we found a clear one. So here are the trial average responses of these two inhibitory neurons to the 4 familiar stimuli. The one on the right that makes a lot of connections, it is selective, meaning that it responds differentially to them. But the one on the left that does not make many connections, it is not selective. It responds about the same to all 4 odors. So this holds across the entire population, where to capture it, we defined a selectivity index, that's the x-axis here. And indeed, inhibitory neurons that are more selective across the familiar odors, on the right on this plot, form abundant connections within the network, while those that are more selective to the novel stimuli are less connected. Critically, this was an experience-dependent effect, as we don't see it in naive animals. So inhibitory neurons that respond differentially to familiar stimuli make more connections. But what came first, differential responses or connectivity? Because either the most densely connected inhibitory neurons somehow became differentially responsive, or those that already happen to be selective through connections. And we don't know the answer to this question now, and I'll come back to this at the end. But before I do, I'd like to tell you about some modeling we have done to explore how a network connected this way behaves. So for this, we developed a simplified feed-forward model agnostic to whether it's one or the other mechanism. At initialization, both inhibitory neuron connectivity and selectivity were assigned at random, and then we rewired the network. We took inhibitory neurons that happened to be highly selective to the familiar stimuli, say this one, and we added outgoing connections, while we removed connections from non-selective neurons. So now that the model has the same relationship between inhibitory connectivity and selectivity as we saw in the data, it also shows the same enhanced discriminability for familiar stimuli. So how does this work? Because one easy way to increase discriminability would be to increase inhibition across the board and sparsify the activity. But this is not what is happening in our data, because responses in the data are equally sparse for familiar and novel odors. So somehow this network is decorrelating without sparsening, which has the advantage of preserving the signal-to-noise ratio. To understand this, we went back to our model, which turns out makes a precise prediction about the mechanism producing decorrelation without sparsening. And it has to do with inhibitory input currents onto excitatory neurons. More precisely, that after this rewiring, the variance of this current across familiar odors will be larger than across novel odors. In the model, we can see this increase simply by measuring the inhibitory input currents, and here it is compared to shuffled wiring. In the data, we can estimate the currents based on the activity and on the inferred connectivity. And remarkably, our model prediction was verified in the experienced animals, but not in the naive. So this is an example of the kind of structured inhibition that Julijana was describing earlier, but in this case, it makes the excitatory responses more diverse and higher dimensional, both in the model like in the data. So to summarize, we found an experience-dependent reorganization of the piriform that depends only on one factor, how differentially modulated inhibitory neurons are by recently experienced odors. And other factors didn't seem to matter, including the selectivity of the excitatory neuron on the other side of the synapse. And I'm happy to take questions about this. So we proposed that such reorganization might implement a central component of perceptual learning, improving the network discrimination of the stimuli that the animal is currently dealing with. But now back to these two alternatives. So this may be a consequence of an increase in selectivity or of an increase in the number of inputs and outputs. And to get to the bottom of this, we need a longitudinal observation. And we are beginning to do that. These are just the early days, but here's an example of one chronic recording over more than 10 days with 1 hour of acquisition every day. Here's a connected pair of neurons on day 0 of the recording in which the connection stuck around for a couple of weeks. Here's a different pair of neurons, same recording, in which we are seeing the emergence of a connection. And here's a third connected pair, where a connection seems to be decaying. And ultimately, this is the kind of signal we need to resolve the chicken and egg question that I just posed, and more generally, to discover learning rules in a living and learning brain. If you're interested in working with the three of us in these kind of questions, I'll be starting a group at a new theory center at UChicago this fall. We have an announcement for postdocs on HOVA, and the experimentalists, Andrew and Carl, have just started their labs at Northwestern and at the Allen Institute, respectively. Thank you.
Yeah, we have just time for one quick question.
Yeah, yes, please. Quick question. Hi, over here. Hi. Oh, yeah. Hi. Yeah. So I have a question about your method for identifying putative connections. How big of a factor is the latency between the two neurons? It seemed like all of the examples you showed had a pretty fixed, like 2 millisecond latency. Do you exclude, for example, spurious connections that have a very high correlation but no latency?
Great question, thank you. So we typically, those connections that are more nearby have sharper peaks, but we have a large heterogeneity of connections. So here you can see all the heat map of all the cross-correlograms that we defined as connected. And you can see that there is a heterogeneity of delay in the peak. And actually, you can correlate the peak here in the y-axis with the distance, estimated distance between the two neurons. What you see is a very nice correlation between the two. And actually, if you estimate the conductance velocity of the axon from this, you get a value which is compatible with what has been measured in the piriform.
And do you have an exclusion criteria for a minimal latency?
No, we don't. I mean, we don't look at peaks that happen before 0.5 milliseconds. Yeah.
Okay. All right. Thank you.
Thank you.
Okay, let's thank Sam again. And the next speaker is Huszar, also from Columbia University. And the title of his talk is Learning Dynamics in Development Defined Microcircuits is Rooted in Inhibitory Connectivity.
All right, thank you so much for that introduction. So before I begin, I just wanted to acknowledge my co-authors at NYU and at Duke, and especially highlight Artem Kirsanov, who did a lot of the work on the analysis that you'll see. So the hippocampus is important for learning and memory by constructing cognitive maps, which are internal models of the outside world. And this is thought to depend on very complex microcircuits that are very challenging to access in vivo. But we know that distinct microcircuits arise at different time points during development, which means that we can use development to access a complex microcircuit that might have functional relevance for cognitive mapping. So we set out to do this by labeling neurons that were born on the same day during embryonic development. So briefly, this involved timed virus injections into mouse embryos that allowed us to deliver channelrhodopsin into a set of neurons that were born on the same day in the hippocampus, specifically in regions CA3 and CA1, where previous work has shown that these developmentally related neurons exhibit preferential monosynaptic connectivity. So when these animals become adults, we can record their neural activity in CA1 and CA3, and we can identify a subpopulation of developmentally related neurons that is embedded into this larger microcircuit. And what we observed is that these neurons exhibited higher spike time correlations during running, but also during sleep. And they also exhibited convergent connectivity on inhibitory interneurons. This is something I will elaborate on later on. So what we have here is a developmentally pre-configured microcircuit that exhibits connectivity and constrained connectivity as well as coactivity. And we were interested to know if the activity in this microcircuit is simply a passive architectural constraint. You can think of it as the fumes of development, or if it might actually have functional relevance, perhaps serving as a building block in a cognitive map. So we set out to test this by monitoring the dynamics of developmentally related neurons throughout spatial learning, which is an important function of the hippocampus. So we trained these animals on a spatial learning task, which involved finding a location of 2 new hidden reward locations. So the task was very simple. The animal simply had to run to the rewards, find them every day, and then go back to the waiting box to initiate the next trial. And so across trials, the animal's behavior became very biased to these reward locations. So the animals took more optimal paths to recover these water rewards across trials, so indicating clear spatial learning. And so this learning phase was preceded by an hour-long sleep period that we'll call rest 1, as well as followed by another sleep period, rest 2. And in fact, the neural activity throughout sleep is going to be crucial for our analysis, because it's going to allow us to identify what changed in the hippocampus as a result of learning. So for this, we will focus on sharp wave ripples, which are bursts of hippocampal activity throughout which this system reactivates behaviorally relevant activity. So in doing so, the hippocampus is effectively showing us the learning-related patterns. So if we think of neural activity throughout ripples as living in a high-dimensional state space, the activity in rest 1 ripples occupies a specific region that is distinct from rest 2 ripples. So to identify these 2 regions, we used contrastive PCA that allowed us to identify directions of variance that were common across rest 1 and rest 2 ripples, directions that were unique to rest 1, as well as those that were unique to rest 2. And throughout this presentation, I'll refer to the rest 2 specific directions as the new subspace, since these patterns first emerged in the sleep after learning, and the rest 1 specific directions I'll be referred to as the old subspace. So now we can ask, what do these ripple-defined subspaces tell us about learning? So we take the population activity recorded as the animal is running on this arena, learning the reward locations, and then we project it into the old and the new subspace. This lets us examine what information each subspace captures about behavior. So let's consider neural activity as the animal approaches the reward. We can visualize projections into the top 3 contrastive components and then color each of these projections according to the distance of the animal relative to the reward. And we find that the new subspace contains a lot of information about the animal's progression relative to the reward. And in contrast, there isn't too much appreciable structure in the old subspace. So we quantified this by training a nonlinear decoder that took as its inputs the instantaneous activity in the network to reconstruct the animal's position. And the decoding was done based on reconstructions of neural activity following a projection into the old or the new subspace. So in evaluating these 2 decoders, we found that reconstructing the position of the animal based on the new subspace was more accurate, so reflected in lower decoding errors, and especially near the reward locations. So this suggests that this new subspace encoded the learned reward location. So finally, we're at the point that we can ask about the contribution of our development-defined microcircuits to this learned information. So to address the role of individual neurons in a population code, we borrowed attribution methods from the model interpretability subfield in machine learning. So briefly, for each neuron, we assigned an attribution value, which effectively measures how informative this neuron's activity was for reconstructing the position of the animal at each time point. So for each neuron, we have a time series of these values. You can think of them as time series of decoding weights. And for each pair of neurons, we compute the attribution correlation, which measures how coordinated the information was for each pair of cells from the lens of this decoder. And so we compute the attribution correlation for each pair of developmentally related neurons in relation to neurons that were born on different days. So the 0 on the y-axis here is the attribution correlation for a typical pair of neurons born on different days. And we saw that for new subspace decoders, which captured the learned reward location, neurons that were born on the same day had higher attribution correlations, meaning that they contributed information to the decoder in a coordinated manner. And in contrast, these neurons made uncoordinated contributions to the old subspace decoders. So this pattern was appreciable for pairs of developmentally related neurons in the CA1, but also for pairs across the CA1, CA3 subfields, but not within the CA3. which we thought was interesting because the CA3 provides inputs to the CA1. So in other words, developmentally related neurons encode similar reward information in the new subspace, and this learned reward encoding first arises at the level of interactions across these two hippocampal subfields. Now, I mentioned at the beginning that neurons born on the same day exhibit convergent connectivity with the same interneurons. So we were interested in this microcircuit motif. So to identify excitatory inhibitory connections, we computed the spike time cross-correlograms between each pair of pyramidal cells and inhibitory interneurons. And for many pairs, we detected instances in which the inhibitory cell spiked reliably within milliseconds of the pyramidal cell spike, which we took as evidence of putative monosynaptic connectivity. So this is a very similar approach that was explored in detail in the previous talk. So we're using a complementary method, but that detects connections based on the same idea. So we detected many such connections in the CA3, CA1, so lots of EI connectivity involving these two subregions. And then for each pair of pyramidal cells in the dataset, we computed the degree of convergence on inhibitory cells. And this was a metric that varied dramatically in the dataset. So there were pyramidal cells that had almost no interneurons in common, all the way to pyramidal cells that targeted almost the same set of interneurons at the level of their output. And so we took the extremes of this distribution and considered them neurons born on the same and different days. So using connectivity with interneurons as a proxy for developmental relatedness. And as for the analysis for developmentally related neurons, we computed the attribution correlation now relative to neurons that had low convergence on the same interneurons. And we found that for new subspace decoders that captured the learned reward locations, neurons that had high degree of convergence on inhibitory interneurons had higher attribution correlations, meaning that they made coordinated contributions to the decoder. And these neurons made uncoordinated contributions in the old subspace, suggesting that they did not represent the same information. So it's a very similar pattern of results we saw for developmentally related neurons. And this was apparent for pairs across the CA1, CA3, but also within CA1 and not within the CA3. So in using convergent connectivity on interneurons, we revealed a strikingly similar pattern of subregional contributions to the learned reward encoding, as we saw with developmentally related neurons. So to summarize, we saw that embryonic development produces microcircuits with constrained connectivity, and the correlations of these neurons are maintained throughout learning, but also reactivate throughout sleep later on. And finally, that these developmentally pre-configured microcircuits play a unique role in encoding a new memory for an important location in the environment. And so we suggest that this may in part be due to these neurons' privileged connectivity with the same set of interneurons. So all this can be summarized with a simple take-home that neurons that are born together will wire together, fire together, and learn together. And with that, thank you so much for your attention, and I'll take any questions if we have some time.
Yeah, we do have time for a few questions. Maybe here. Yeah.
Thanks for the great talk. I was curious, did you look at the replays of the old and the new neurons in your analysis by any chance?
Yeah, so basically these, I would say that we used, the existence of replay allowed us to define these subspaces because we basically identified them based on comparing the neural activity in sleep sharp wave ripples before learning and after. So the old subspace is enriched in ripples before learning, whereas the new subspace is enriched in ripples after learning. And so then the hypothesis is that these subspaces are different in encoding what the animal learned about, which is the reward locations. And so that's how we ended up motivating the meaning of that subspace. Yeah, so that's exactly what I think we're capturing.
Okay. If there's no further questions, let's thank the speaker again. And we will be back in half an hour after the coffee break.